this webinar will be recorded and it will be made available via the SBTS and TBTS social media, in particular our YouTube sites. We also have everyone on mute as the presentation is actually a recording, but please send in any questions via the question and answer function or the chat function of Zoom, and Matt will answer them at the end. Please feel free to upvote questions that you in particular really want to hear the answers to.
Yep.
I'm just going to open up a second question there. We've got 3 to answer here. Matt, do you want to have anything to say before we kick off?
Just to say thanks to everyone for joining in on this. I think we've got pretty good numbers along. I very much see this as a bit of a pilot. I think it's a pretty efficient way to get information out. We'll see how we go today, but I would like to think this is something we could do a little bit more regularly, certainly, than we've done in the past. So we'll do our best to get to all of the questions. If there's any left over at the end, I'll happy to get in touch with people. But we'll play this recording, which I have to say is my first attempt at this and not the most comfortable 45 minutes of my life. But we will play it through and we'll talk to people at the end.
Cool. Right. Just getting a few final answers in. And we will kick it off. So we will put ourselves on mute and we'll play the presentation, and then yeah, as we said at the end, we will attempt to answer the questions, or Matt will answer the questions for you.
It'll be a team effort.
Cool. Right. So if I close polling and share screen onto that, which should come up. Oh, and share computer sounds rather important, so you can hear Matt when he's talking. And here we go, everyone. Have fun.
Good afternoon. Thank you for joining in on this unusual version of a conference, and thanks also to the organisers of BIF for asking me to come and talk about some work we've been doing in Australia, over the last 20 years or so. I was asked to talk about new reproduction traits that we're including in our genetic evaluation, how we're working those into industry, and how people are taking advantage of those to accelerate genetic progress for these economically very important traits. So there are 3 main areas I'd like to cover over the course of this presentation. The first is looking at how these new reproduction traits were developed, and that's going back some time into the research undertaken as part of the beef CRC. Secondly, I'd like to look at how these traits have been implemented in industry, and that'll be a fairly brief summary of the Repronomics project, and then a closer look at work we're doing within a specific Brahman seed stock herd, bringing these hard to measure and quite expensive traits into industry. Finally, I'd like to talk about some more recent research in temperate breeds, Angus and Hereford specifically, that's examining the genetics of reproduction for those more southern adapted sorts of animals. So firstly, just a bit of a look at how these reproduction traits were developed. And I really see this as having the aim of creating opportunities for breeders to select to improve reproductive rates in tropical beef cattle. So as I kind of pointed out earlier, The Beef CRC was a multidisciplinary collaborative research project. It involved lots of scientists looking at lots of aspects of productivity in the beef industry. A key component of it was that it's industry supported, and all of the CRCs required that they have industry funding which was matched by the government to allow the research to happen. And in this case we had significant support from industry to focus on tropical genotypes, and it very quickly became clear that reproduction was a key aspect of what we needed to look at. As I said, it was about a 14-year progeny test. As part of the first round of that enterprise, we looked at steer productivity, which included carcass traits, and we were also looking at heifer reproduction traits in their half-sib sisters. In the second phase, we moved on to look at cow rebreeding and lifetime reproductive performance. So just a quick look at the design of the experiment. It didn't involve massive numbers of animals. We really had the philosophy that we wanted to record everything we possibly could on these animals, and that being the case, we were not able to do it on a massive scale. But over that period, we looked at the performance of around 1,000 Brahman females and about 1,100 tropical composites. Those were all bred in co-operator stud herds and then contributed to the CRC where they moved to research properties at weaning. The breeding was designed. There were about 50 Brahman size and 50 tropical composite size, and they were allocated to ensure genetic linkage across the herds of origin and the properties where those females ended up. So female reproduction was recorded intensively in these animals, and as we said, their half-sib brothers were finished in the feedlot to about a 540 kilo live weight specification. So just a bit on the management of the females involved in these projects. So as we just saw, they were located at 4 research stations around Northern Australia. Heifers were first mated as 2-year-olds, which is very much standard for the industry up there. A little less standard, we enforced a fairly tight 3-month mating period. There is plenty of enterprises that leave bulls out all year round up there, but in this instance, we wanted to, push them reproductively, and a shorter joining period allowed us to do that. They were managed under decidedly commercial conditions. Feeding only happened, to avoid any critical body condition situations arising, which certainly did happen over the years that CRC ran, but there was never intent that these animals be maintained in anything but commercial conditions. We also made sure we gave females the opportunity to express as much of their genetic differences for reproductive performance as we could, which meant that we retained all of the females in the experiment unless they failed to wean a calf in consecutive years. So I think this is a key part of this aspect of the presentation and is central to what we will look at going forward. This is just looking at the traits that we've developed to allow us to describe female reproduction. For female traits, they exploit ultrasound scanning to look at ovarian function. It's a process that's very similar to preg testing, but the technician has an ultrasound probe and are able to image the ovaries fairly carefully and identify whether an animal is cycling or not. This is just a comparison of, on the left, a prepubertal ovary showing a lot of small black follicles but no apparent corpus luteum, where on the right we see an ovary that is completely dominated by a single large CL. Once you get a bit of practice with it. They are structures that can be identified fairly easily, but doing so quickly and accurately takes a lot of practice and a lot of skill. And it's something we've been thinking about as we go forward with this, building a bit of capacity in the industry for people to provide this service. So the first trait that we defined was age at puberty, and it was based on serial ultrasound scans to observe estrus, which meant that scanning started in animals at around 200 kilos, which wasn't long after weaning for many of these cohorts, and carried through for each heifer until a corpus luteum was identified. And that meant for some animals having up to 15 scans, before they identified that the animals were cyclic. The next key trait was one that was measured in lactating females, and again use this ultrasound scanning to identify when a lactating female recommenced cycling after the birth of a first calf. It was calculated as days from bull-in, the start of natural mating, until that first CL was detected. And by the end of the CRC period, we had about 60 records on animals that stayed in the experiment for their entire lives, which meant we had more than 350,000 scans collected over that 12-year period or so. And this is the first of the results I'd like to share from this beef CRC research, and these relate to cow lifetime weaning rate, which is just the number of calves a cow weaned divided by the number of mating seasons that she was in the experiment. I think the key numbers to look at here are the average for Brahmins at 0.62 or 62%, and for tropical composites at just under 80%. And these low reproductive rates are really what motivated a lot of the work done in this project. We've also got the range in sire EBVs for the size of these females, and in Brahmins that went from -11% to +16%, which means there was a difference of 27% in the weaning rate between the best and the worst sires. Like a lot of reproduction traits, it's not especially heritable, around 0.1 for both genotypes. And that low heritability, combined with the extreme economic importance of these traits, is a lot of the reason that we were pursuing the traits we'll go on to have a look at in just a minute. In contrast, our age at puberty trait was extremely heritable in the animals evaluated for this study. In Brahmins, it was 0.6%, 60% heritable. In tropical composites, 50% heritable. There was also large genetic variation for the trait within those genotypes. And this plot's just a demonstration of that for the Brahmin females. So the horizontal axis there is age in days at which females displayed their first corpus luteum, were pubertal. And the y-axis is just the number of females that were pubertal at those ages. So this is a distribution of age at puberty in the Brahmin females. And we can see with mating at around 2 years of age, there's only about half of those females cycling as they go into their first mating season. Given that we only applied a 3-month mating period, beyond the end of those 3 months, there's still 13% of animals which failed to reach puberty. And that's going to have, a dramatic impact on the potential for high weaning rates to be recorded. Of those which did cycle, about 90% conceived and 72% weaned a calf, which gave us our overall weaning rate of about 60%. I think the the key result here is with this high heritability and large genetic variation, there is huge opportunity to apply selection to improve age at puberty in Brahman females. And that was certainly the case for tropical composites as well. For the lactation and estrus interval trait, we also saw high heritabilities. The composites had a heritability of 30%, the Brahmins 50%. Again, large variation within these genotypes, which means we've got lots of opportunity to identify genetically superior animals. I think when discussing these two traits, it's worth pointing out that age at puberty only directly influences the first mating outcome of females, where this lactation and estrus trait bites every time animals go through the breeding cycle. So as far as economic impact, lactation and estrus interval is probably the more important of the traits. So this is just an example of estimated breeding values for the Brahman sires that were involved in this study. And it's worth for this audience pointing out that the EBVs we publish in Australia are descriptors of the animals themselves and not the differences we expect to see in their progeny, which just means that EBVs are twice the value of an EPD. And what we saw was that for these Brahman sires, there was a range, even if we cut off the outlier at the bottom of the page, of better than 200 days between the top and bottom sires. This means that the best size progeny will have a lactation and estrus period 3 months shorter than those of the worst sires in this list. And it's worth pointing out, as we look down this list of beef CRC sires and their lactation and estrus EBVs, that the more negative the breeding value, the more favourable the reproductive outcomes. So we're wanting lower rather than higher EBVs. It's also true that with that level of variation, under average conditions, there will be sires whose progeny are essentially not going to cycle within a short 3-month mating period. And we certainly did observe that in those bulls. You know, we identified heifers that were the progeny of a couple of those particularly bad bulls for lactation and estrus, and they didn't cycle before they weaned their calves. The other thing that happened as part of this project was a fairly close look at opportunities to improve our understanding of the genetics of male reproduction. So as part of the third round of the CRC project, the male progeny of the cows we've just been talking about evaluating for age at puberty and lactation and estrus interval were retained as bulls. They were semen sampled at 12, 18 and 24 months of age. They had sperm morphology assessments done, which involve looking at 100 cells for each animal, identifying any non-viable cells and classifying those into different specific abnormalities. But the key number we'll look at here is percent normal sperm, which is just the proportion of viable cells within that sample. A key result for this trait is that variation in general, and certainly genetic variation and heritability, were very age dependent. This table is a demonstration of the additive variance and heritabilities for Brahman and tropical composite bulls for samples collected at 12, 18 and 24 months of age. The first thing to jump out from these results was that Brahmans produced almost no viable samples as yearlings, and it was only at 18 and 24 months of age that we got useful samples from them. But there's a big difference in the level of genetic variation between those two sampling periods. Almost 200 at 18 months of age and less than half of that at 24. And the heritability is correspondingly 0.3 at 18 for Brahmins, 0.2 at 24 months of age. For the tropical composites, interestingly, they showed the greatest variation and the greatest heritability for the trait at an earlier age, at 12 months old. And it's quite consistent with what we see in these sorts of animals running under commercial conditions in Northern Australia. Brahmins become sexually active a lot later than their composite counterparts, and certainly a lot later than British bred sort of cattle. One of the really important questions that we wanted to address in looking at male reproductive performance was what information it could give us about their female relatives. And these are genetic correlations of Brahman bull percent normal sperm with female age at puberty and lactation and estrus interval. So as we saw in the last slide, at 12 months of age, we're getting no information from these young Brahman bulls. But at 18 and 24 months, we're seeing consistently negative genetic correlations, which is favourable. That explains a relationship of higher percent normal sperm with lower age at puberty and lower lactation and estrus interval, which are both desirable. And those correlations are strong enough that we can exploit these measurements as indirect descriptors of female reproductive performance. Really pleasingly, it's a measure we can take in selection candidates. So we can take a semen sample from a young Brahman bull, do a morphology test on it, include those results in a genetic evaluation, and via these correlations get information about the reproductive performance of their female relatives, as opposed to having to mate those bulls, do a progeny test on them, get their female progeny, mate them, see how long it takes them to become pubertal and how long it then takes them to conceive when lactating. So it definitely shortens the time for data to come in on these key traits. So I think just to wrap up the CRC element of this, I think key results from this work was that we had developed some really accurate descriptors of female reproduction, and that those were, for a reproduction trait, very heritable. It means we had tools which would allow us to make rapid genetic progress for these economically important traits in tropical beef breeds. We also showed that male traits can be exploited as genetic indicators of these females' descriptors of reproduction, but there's no getting around the fact that these are difficult and expensive and expertise-intensive traits to record. There is a lot of inputs required to get this data, which in the era of genomics makes them prime candidates for recording in a reference population situation. And that's one of the things we'll look at now. So just a bit of where we went from the end of the CRC before we wrap that up. In our breed plan evaluation for tropical breeds now, we have results for age at puberty and lactation and estrus interval coming into the analysis. They're being recorded intensively in research herds, which are putting data into the evaluations for these breeds. Genomics is being applied to spread accuracy for these traits to related animals. And both of them are contributing to the trait which currently describes female reproduction, which is days to calving. So we don't have separate EBVs for age at puberty and lactation and estrus. They're just driving accuracy via correlations with days to calving for that trait. More recently, we've also introduced percent normal sperm EBVs for Brahmins, and coming soon for Santa Gertrudis. And again, it allows breeders to select to improve percent normal sperm directly, and at least as importantly, it serves as a correlated trait with female reproduction. So the next couple of areas I'd like to cover are how we're taking these complicated, expensive traits from research to industry. And the first of those is a very brief look at the Repronomics project. This is a large industry-funded project that's led by Dr. David Johnson, who I think some of you will be familiar with, and is really applying what we learned in the beef CRC on a larger scale and under more commercial conditions. It's still focusing on those same traits, so we're intensively recording heifers for age at puberty. We're looking at lactating first calvers and recording lactation and estrus interval. Unfortunately, we're not getting any male reproduction from this particular project, but it's one of the features of the next project we'll look at. The males in this case are being steered and are going to sorter to give us some carcass information. The Repronomics project has expanded on the breeds that were involved in the CRC. So we've still got purebred Brahmins involved, but we've also included Santa Gertrudis and drought master animals, which weren't in the initial CRC research. There's also plans for crossbreds and their performance to be evaluated in the next phase of this project. And that's likely to start in the next 3 or 4 years. And key to this work is that as quickly as we can generate this information in this project, it's being included in the respective evaluations for these 3 breeds at the moment. So we're getting this data as quickly as we can, and it's driving EBV accuracy within the commercial evaluations for these breeds as quickly as can be done. The next aspect of getting this technology out into the industry we'll look at is some work we've done with a privately owned stud producer in Northern Australia. The property is called Kaiuroo. AGBU has had a interaction with them for 5 or 6 years now. The Kaiuroo enterprise is an aggregation of 5 neighbouring properties in the Fitzroy River Basin of central Queensland. The map on the right there just identifies the location of the property relative to Rockhampton on the coast, which is kind of the centre for tropical beef production in Australia. Combined, the properties make up 34,500 hectares, 600 of which are irrigated Lucina. Lucina is a leguminous shrubby crop. It produces high energy, high protein feed, but is difficult to manage. Stock-wise, there are 1,000 Brahman stud females run on the 5 properties, and about 5,000 commercial cows. Commercially, the herd is targeting an organic market. It's a market that has reasonably high costs of production and tight specifications around what they accept. They're targeting turn-off weights of about 480 kilograms, and there are some minimum requirements around growth rate to meet these market specifications. That was challenging for the business when it started, and AGBU was approached in 2014 to try to evaluate the genetics of the herd and make any suggestions that could help them improve compliance for that market. So following a review of the Kaiuroo breeding program, It was identified that, like a lot of breeders of tropical cattle, low reproduction rates were a key limiter of productivity and profitability for the herd. Because the herd had been so well evaluated, we were able to benchmark them against the Brahman breed pretty accurately and identify that they were below breed average for days to calving and scrotal circumference, and about breed average for our growth, fat, muscling, tenderness and temperament traits. The review concluded that a targeted breeding program could improve profitability for the herd. Intensive recording within the stud presented opportunities to increase rates of genetic gain for these key reproduction traits, and the production system and market that they were targeting warranted creation of a custom selection index. So in 2015, we implemented an ovarian scanning program measuring age at puberty in all the young heifers and lactation and estrus interval in lactating first calf females. And all of the bulls were also semen sampled for sperm morphology testing at 18 and 24 months of age. This was an expensive process, and we approached the industry funding body MLA to see if we could get some assistance with the costs of this exercise, because we could clearly demonstrate that the data that was coming from this recording was of clear benefit to the broader industry across the entire Brahman breed. Data was going into Brahman breed plan and was benefiting any animals that were related to those recorded in the Kaiuroo herd. Importantly, it was also the only source since the end of the beef CRC of percent normal sperm data coming into the Brahman evaluation. Thankfully, we were successful in attracting that funding, and we got some support to help us with the cost of the recording program, as well as genotyping of all of the males and females that we were evaluating as part of the project. We were also careful to make sure that the project was well linked to the Repronomics project by exchange of genetic material. So the data coming from this can also be analysed as part of that project. So to date, we've collected about 700 records of age at puberty in heifers, about half that in lactating first calf females to describe lactation and estrus interval. About 725 bulls have been evaluated for percent normal sperm, and all of these animals have been genotyped with a custom Bos Indicus 35K chip. So far, we've evaluated the progeny of 70 bulls over the 3 years of the MLA project. That's been a total of 7,100, those sires have over 7,000 progeny represented in the broader Brahman evaluation, and 26 of them have progeny outside of the Kaiuroo herd, which totals almost 2,500 animals. As a result of the recording that's occurred just in the progeny of those 70 bulls within this project, we've seen an increase in the accuracy of percent normal sperm EBVs of about 30% across all 70 of those sires. Obviously that fluctuates from animal to animal, and we have to acknowledge that we were starting from a pretty low base for some of these bulls, but an average increase of 30% gives people much better opportunities to apply selection for the trait and do so accurately. Our female recording has resulted in increases in days to calving EBV accuracy in the order of 7 to 15%. This was already a herd that was recording the trait reasonably well, so we're just creating increased spread in the EBVs for animals related to those females recorded as part of this exercise. They're also getting a fair boost in female reproduction trait accuracies from the Repronomics project, given the links that we've established between those two projects. But the effort within this one herd has spread opportunity to apply accurate selection for reproduction traits to a lot of breeders around the Brahman breed. Finally, and fairly quickly, I'd like to just bring you right up to date with some work we're doing currently in temperate breeds. We've done all this work in the last 20 years looking at these traits in tropical breeds, and this is just playing a bit of catch up really in temperate breeds to help us understand the genetics of these traits in Southern Australia. So the objectives of this work was to apply the serial scanning methods that we developed in the beef CRC to look specifically at age at puberty in temperate beef heifers, and that was a group of Herefords and a group of Angus females from seed stock breeders. We wanted to quantify the variation in age at puberty in the current population, just see if there was any differences. We really were going into uncharted territory with this. We weren't sure what we would see, and certainly we do not face the same level of problem around reproduction in temperate breeds that we see in the tropicals. And once we've collected these records, finally to look at the genetics of these traits, whether there's genetic variation, whether it's heritable, and whether there are any genetic relationships with cow body composition. So the animals involved in this study were from 7 Angus herds and 3 Hereford herds. They were all herds that have a long history of submitting good quality data to breed plan. They were registered with their respective societies, and as an absolute minimum, we needed them to be contributing dates of birth and genotypes with pedigree recorded. All of them were doing that, and all of them were doing far better than that. The traits we were looking at is a binary trait, 1-0, whether animals were pubertal into mating or not. We also had age at puberty as defined for the beef CRC, just the date at which we observed their first corpus luteum minus their date of birth. And for any animals that weren't cycling, we also created a penalised trait, which was the maximum age at puberty of an animal's contemporary group plus 21 days, one cycle, for any animals which failed to display a CL up to mating. And in this case, mating was synchronisation for AI. So all of these, we had to stop recording once these animals were synchronised. Obviously, synchronising estrus completely removes our opportunity to record it as a natural trait. While we were ovarian scanning, we were also recording information about female growth and body composition. So we're getting weights, measuring hip height, using ultrasound to measure fat depth, and giving the animals a body condition score. That's 1 being poor and 5 being in extremely good condition. It's worth pointing out that the technicians that we used for this work were extremely good, experienced ovarian scanners, but they were not accredited breed plan carcass scanners, and most of them hadn't had a lot of experience condition scoring. So I think the key result to come out of this, and it was one that we really were not expecting to see, was that only 52% of heifers were pubertal as they entered their first mating, as they came in for synchronisation for AI. So these results are just some averages and standard deviations for the Angus and Hereford animals that we scanned for this project. And we've got results for their age at puberty, the penalised age at puberty with and with such a high proportion of animals not pubertal, this trait then becomes a really key trait. There's also averages for the proportion that became pubertal. We can see we got records on almost 3,000 Angus and almost 1,000 Herefords. And as we said, they're only about 52% pubertal going into mating. Age at puberty for those animals that were cycling was very close to a year old for both breeds, and with the penalty applied for those that didn't cycle, it got out to almost 400 days. The next thing that was interesting was that on average these females weren't in bad shape. We really can't blame condition on their failure to show any signs of estrus up to their first mating. This again is just some raw averages of their growth and body composition as at their first scan, so that's post-weaning, and their final scan into mating. Post-weaning animals were just a little bit under 300 days old, a little bit younger for the Herefords, into mating around 400 days. Weights, they were around 300 kilos at their post-weaning scan, 300 and almost 70 for Herefords, 370 for Angus, 340 for Herefords. Hip heights were almost identical. Condition scores are a bit telling here, and both at 2.9 for Angus, 2.6 for Herefords, are describing animals that weren't in bad shape, although it's worth pointing out that there was variation in this. There were certainly animals within herds that were not in as good a condition as some others. Fat-wise, 5 mils of fat post-weaning for the Angus, going up to 7 into mating, and almost exactly the same result into mating for Herefords. So those results show that on both at the raw level, for both puberty and growth and body composition traits, Angus and Herefords showed almost no differences at all, which was pleasing, I must say. This was not set up as a breed comparison. It's inevitable that people look at these results that way. There's no capacity to pick differences based on the design of this experiment, but it was nice to see that they're pretty much the same. This is just a demonstration of the proportion of animals within each breed, that's red for Herefords, black for Angus, at each scan. On average, our Angus got 4 scans from post-weaning to mating, Herefords only got 3. But we can see that that trend is very consistent across both breeds and describes a pretty consistent rate of animals becoming cyclic over that period. The big question in all this is, do we have opportunity based on these measurements to make useful selection decisions? And really pleasingly, age at puberty in both breeds was highly heritable for a reproduction trait. So if we just look within the animals that had a CL up to their first mating, the trait was almost 30% heritable, 0.26 for Herefords, 0.27 for Angus. The penalised trait, which becomes the go-to trait for most of our subsequent analyses, about 10% more heritable, 38% for Herefords, 37% for Brahmins. And even because it's so close to 50%, analysing the binary percent pubertal trait gives us opportunities to make genetic progress for the trait. 36% heritable in Herefords and 32% in Angus. Finally, one of the key questions we wanted to address in this early work in Bos taurus animals was whether there were genetic relationships between body condition, that's growth and body composition traits, prior to mating, and their performance as far as puberty went. And I think it was interesting to observe that there were not strong genetic relationships between either their weight, their height, how fat they were, or their overall condition score. And this table just presents the genetic correlations in the right-hand column between the penalised age at puberty trait and these four metrics of growth and body composition, weight, height, fatness, and condition score. We can see that all of them describe a relationship of higher growth and body composition with lower or younger age at puberty, but that none of them are especially strong. The strongest relationship was actually with condition score at -0.26, but it's not suggesting that there are causal links at the genetic level between these traits, and certainly, unfortunately, no real opportunity to exploit these much more easier descriptors of body condition as correlated traits with age at puberty. So I think the next steps in this work, we need to record the trait in naturally mated females. When I designed this project, it was, on the back of massive anecdotal evidence that these breeds do not have major problems with reproduction. Certainly conception, calving, weaning rates are pretty good for Angus and Hereford heifers. The fact that we only saw about half of them cycling into mating means that we're going to have to continue scanning beyond mating, that we can't synchronise for AI, and we have to observe variation as bulls are introduced to the herd and see what happens between then and the end of that mating period. To that end, there's a new project just in development that will mate 2,000 females of numerous breeds annually, and all of those will be evaluated, or the progeny of all of those as they come around will be evaluated for age at puberty and lactation and estrus interval. These are all females that are sourced from well-recorded herds, and we've got as many breeds as we could represented in that study. So there will be Angus, Herefords, Shorthorns, Wagyus, Charolais and Brahmins involved, and those Brahmins will give us linkage to the Repronomics project up north, which will really add to the value of this work. So just to summarise, I think over the course of the research that we've described in this presentation, we've been able to develop and prove some methods to accurately describe reproduction in both males and females for tropical breeds, and those traits have been implemented in the genetic evaluation for those breeds. We've developed a framework for reference populations, both in a research-based structure as well as in commercial seed stock herds, and that's been undertaken successfully. In temperate breeds, the research into these traits is at a much earlier stage, but our early results suggest that age at puberty definitely warrants further attention. The fact that we only had 50% of heifers pubertal into mating, I think, means that it's a trait that needs to be included in the genetic evaluation and at least monitored as selection pressure is applied in other aspects of productivity. Clearly for those breeds, we also need to tackle lactation and estrus interval, and that will be the focus of future research in those breeds. Thank you for your attention, and I look forward to taking any questions that you may have.
Well, thank you, Matt, for that. If anyone has any questions, please feel free to send them in via the chat function or via the Q&A area that is down the bottom of your Zoom screen there. I see you got anything you want to say, Matt, straight afterwards before we get into the questions, or?
Well, very briefly, I think, Yeah, clearly that was designed for an American audience. I think a lot of people here will have been reasonably familiar with some of that work, but I hope there was some new content for an Australian audience, and certainly the work we've done in the Trans-Tasman project, we haven't been able to talk about a lot until now. Those analyses, have only been completed recently, and there are some really interesting results coming out of that work. And I'm pleased we'll be able to carry it on as part of the Southern Multi-Breed project. That's just in its early stages now.
Cool. Right, I see we've got a quick question here from Felicity Hamlin-Hill. In the CRC, the terminology used was PPAI and various versions for repeated for 1 and 2 there. We now to officially change the name that we use in our workshops to lactation and estrus.
We officially changed it quite a while ago, and this was Jono's idea and a good idea. The PP and PPAI refers to postpartum, and we were not scanning, from the birth of their calf onwards. We started scanning at mating. So there's that period from when a female calved to when the bull went in, when we don't know what was going on. For obvious reasons, you do not want to muck around with them at that stage. And that's why we changed the name to lactation and estrus interval. We didn't want to suggest we were scanning them or getting them in to scan them when they had little baby calves at foot.
Cool. Still lacking a few questions. We've got another couple that have come in via email to us. Do we still need days to calving records to be recorded with all these new traits coming online?
It's a good question. My initial feeling is that of all the traits we record and analyse in breed plan, days to calving is probably one of the easiest to measure. It does not take a lot of work to get that information into breed plan, and I certainly think that if people are interested in describing reproduction in their herd, that's probably the simplest way to do it. Certainly by comparison, these are complex traits, but by recording them in reference, and allowing genomics to spread that around the various breeds, it creates an opportunity to increase accuracy of our days to calving EBVs. And I think that's where we'll see these used most, in highly recorded reference populations, and then augmenting the accuracy of days to calving that's contributed by industry. But certainly, if individual herds wanted to tackle these, as we saw, Kaiuroo do up north, that opportunity is there. It would certainly be a very quick injection of accuracy to their reproduction EBVs.
We've got another question here. To improve the age of puberty in our females, should we be selecting Brahman bulls that have high normal sperm at 18 months?
Well, I think that's what those results show. There's moderate, at least, genetic relationships between improved percent normal sperm and improved female reproduction. So it's wired into the breed plan evaluation that way, so that we're getting information about female reproduction from those percent normal sperm EBVs. So if your goal is to make selection decisions to improve female reproduction, the best way to do that is always going to be selecting on the basis of female reproduction EBVs. But you can do that in the knowledge that we are exploiting percent normal as a correlated trait.
We've got another question in. With respect to only 52% of the Angus and Hereford heifers cycling as bulls go in, do you know the ultimate pregnancy rate on those animals?
We're just getting that information through now. It takes a long time for that to filter through breed plan, but as as we expected, reproduction rates in all of these herds are pretty good. Certainly we're not seeing half of them failing to calve. So there's clearly a spike in animals becoming pubertal and becoming effectively reproductive, either via synchronisation for AI or the introduction of bulls into the heifer herd. So I think we're not combating serious reproduction problems in these breeds, but I think the fact that that number is there would be motivation for me at least to think about including age of puberty in the evaluation for these breeds, and that's something we'll be working towards over the next few years.
Right. Are you looking to use collaborators, cattle and research stations for future temperate research, and are you looking for collaborators?
We will always say almost definitely yes. The Southern Multibreed project is going to involve heifers and first calvers on research stations around Australia, and they're specifically temperate breeds. So we will be able to evaluate reasonably high numbers of those animals over the next few years. If people would like to get involved in that work and they're not on those research stations, then I think that's certainly something we can talk to Brad about in the design of that project.
Got another one over here. Can you clarify where the scrotal size traits with the research into AP, percent normal sperm, and lactation and estrus interval?
Yep. So scrotal circumference has always been present as, like percent normal, a measure we take on males to give us information about both male and female reproduction. The correlation that we've got wired into breed plan 4% normal with days to calving is not especially high, but at least it's in a favourable direction. One of the great advantages that percent normal brings is that correlation is a bit higher, so we can exploit better information about percent normal as it relates to lactation and estrus and age of puberty than we can scrotal circumference and its relationship with days to calving. But they're all traits that are favourably correlated with each other. We're just finding that percent normal is a little bit better than scrotal is. Another thing we find with scrotal is, just like percent normal sperm, its value as a descriptor of reproduction is age dependent. And if people leave measuring scrotal too long, the key correlation we observe is with weight rather than with reproductive outcomes. So it's important to make sure if you're recording scrotals that you do that at the right time, and that's information that you'll be able to get from breed plan for your respective breeds.
Next question is, has there been any initial work on semen quality in the Taurus bulls and these new novel fertility traits in Angus or Hereford?
Excellent question. It is at the top of my list. I really want to get out there and get some semen samples on Boss Taurus bulls. The ones who are related to the females that we will be evaluated as part of the Southern Multibreed project are obvious contenders for that, and that's something I'm hoping we can attract a bit of funding for and get rolling as soon as that project allows us to do that. But I agree, it's an obvious and I think an important question to address as quickly as we can.
We've also got a question, what do you think the effect of epigenetics will be on these traits?
My standard response to epigenetic questions is we don't know. it's very complex to analyse, and just my 2-sentence understanding of epigenetics is that it's environmental factors influencing the genes that are passed on to the next generation. Really, it's a fairly new concept, and it's only been confirmed that it actually exists, quite recently. Exactly how that may impact these traits, we don't know at this stage, and we need a lot of data to be and a lot of understanding of both fixed effects, environmental factors, and the genetics of these traits to pin that down. Boyd has done, as is part of his PhD, a lot of direct research on that, and I'm happy for him to give his opinion on that question.
Yeah, it was after my PhD, but I have done what I quite confident is one of the very few attempts to try and quantify the effects of epigenetics in livestock. Now, I was working with dairy cattle on the project, and we found, yes, that the dam's condition during pregnancy was influencing the effect of her granddaughters. And then you go, all right, why are you not looking at the daughters? That's because, a lot of people go, yes, how well you're feeding the cow is it'll have an effect on the progeny, but is that just the change in developmental processes? A malnourished embryo is going to have effects when that calf is born in later production. So we couldn't, that's just a physiological or nutrition effect. It's not an epigenetic effect, because it's not actually affecting the expression of the genes. So if that effect was then passed on a further generation, we were starting to detect an epigenetic effect with that. And yes, we did. The effect on milk production in dairy cows was like 3 to 6 litres, I think, I'm going from memory, per lactation. So not a very large effect. We had massive numbers that I was able to utilise for the project, so it was statistically significant for those of you who like stats, but in terms of practical significance, I would say no, not important at all. And I keep a close eye on the literature on this topic, and I have seen no other in the last, so I've started working on that 9 years ago. I've seen no other attempts at quantifying it in livestock. So
yeah,
it's, as Matt said, a very, very new field of study. And my gut feeling is, yes, there will be effects. Will they be big? I suspect not. But that's until the studies are done, we can't say anything definitive.
Yep, I think that's the bottom line at this stage. But the more data we collect and the more good quality data, the better opportunity we have of pinning down some of these really complex traits to analyse, and that's always the goal of these projects going forward. So the opportunities to identify these effects will improve with time.
Yeah. Right, it looks like we're actually starting to run out of questions. So if anyone's got any more, they got their last chance to get them in to us now, and we'll see. Otherwise, we will sign off and hope you all have a good day, and we all get out of this COVID lockdown soon enough. I mean, I can't even hug Matt for his well job done today. So
thank you.
Yep.
Thanks you all for joining in. This is an interesting exercise, and I think this is a good way to get information out. So yep, we'll see how it develops.
We do have a poll, and we actually have one more question just come back in. So I'll set the poll going, and I'll get Matt to answer the question while I give you guys the polls to look at. So the question is, any comments on the carcass data you collected?
We didn't collect any carcass data as part of the Trans-Tasman work, the age of puberty work we did in temperate breeds. I will let someone else deal with the issues around carcass traits that are coming out of the bins and the Repronomics project, and research associated with that. It is ongoing, and I don't have any up to the minute updates on what's happening with that data or those analyses. So I'll give a pass on that one at the moment, but certainly as that information becomes available, it will be fed out as quick as we can.
Cool. No more questions, I presume, everyone. Well, we'll sign off, and thank you very much for your time today, and hopefully we will see you again. Cheers.